In this experiment, we will use a gas pressure sensor to measure the amount of oxygen produced when hydrogen peroxide is destroyed by an enzyme catalase. The enzyme catalase will be investigated at different concentrations. Connect the gas pressure sensor to any of the ports on top of LabQuest. When the gas pressure sensor auto-IDs, it sets up an experiment for 900 seconds. This particular experiment only needs 180 seconds, so we need to change the data collection length. To do this, tap on the word length to bring up the data collection dialog box. Highlight 900 seconds and change it to 180 seconds, and tap OK. Add 3 milliliters of water to each of the 4 test tubes in your test tube rack. Similarly, add 3 milliliters of 3% hydrogen peroxide to each of the test tubes in your rack. Connect the plastic tubing to the gas pressure sensor with a gentle half turn. Place 1 drop of the enzyme solution into test tube 1. Quickly stopper the test tube, swirl gently, and connect the other end of the plastic tubing to the top of the stopper, and start data collection. Continue to swirl for all 3 minutes of data collection. When data collection is finished, remove the stopper from the top of the test tube. You'll notice that the graph auto-scaled. To determine the rate at which oxygen was produced, tap the Analyze menu and choose Curve Fit. Pressure. From the pull-down menu, next to Choose Fit, choose Linear and tap OK. The slope represents the rate at which oxygen was produced. You will repeat this experiment with different enzyme concentrations. In test tubes number 2, 3, and 4, you will put 2 drops, then 3 drops, and 4 drops of enzyme in each test tube. After completing run 4, you can show all runs on the graph by tapping run 4 and choosing all runs. As you can see, the rate of oxygen production is highest when the concentration of enzyme is highest.